Viruses are everywhere. The first reports of viruses in ants come from about 50 years ago, and since then many more have been discovered. We hunted through the scientific literature and found that to date there's reports of 87 different viruses across 38 ant species. Many viruses have been detected in the red imported fire ant Solenopsis invicta, as people have searched for biocontrol agents for these ants. Also, many viruses have been discovered in the Argentine ant Linepithema humile, to see if these ants might serve as reservoirs for honeybee viruses. In fact, the 5 most commonly detected viruses in ants are more typically associated with honeybees: acute bee paralysis virus, deformed wing virus, Kashmir bee virus, black queen cell virus, and Israeli acute paralysis virus. Honeybees are well studied, which allows for the detection and the identification of their viruses, but we suspect that many so-called honeybee viruses are actually shared widely among Hymenoptera and may spill over, especially between closely related species.
Viruses are ubiquitous, but also diverse in their form. One way to classify viruses is to look at how the genome is organized. Many viruses that infect ants are positive sense single-strand RNA viruses, meaning that their genetic material is made of RNA. RNA viruses are characterized by high mutation rates and are often able to infect multiple hosts, including various ant families. These Argentine ants right here likely harbor a variety of viruses. The same viral strains are often found to infect different hosts in a given area, such as ants and bees, for instance. This further suggests that the viruses that can infect ants also have the potential to jump from one species to another, which can have wide-ranging implications for disease emergence. Invasive species like the Argentine ants are likely to form disease reservoirs, and arrival of exotic species into new regions have been shown to modify pathogen networks within recipient insect communities.
Next-generation sequencing techniques have revolutionized virus discovery in ants. There are 87 known virus species that have been found in ants, many of which were only discovered within the last 5 years, and there are likely many more to be discovered. A lot of virus discovery has focused on 2 pest species, the Argentine ant and the red fire ant. However, the advantage of next-generation sequencing is that we don't need prior knowledge about which viruses to test for, which is a great advantage when testing species that are not well studied. For a lot of viruses that were discovered in ants, we don't know whether they actively infect and parasitize the ant host. Ants may prey or scavenge on infected arthropods and ingest viral particles that are then detected through PCR or high-throughput sequencing. Testing for viral replication gives a good indication that a virus is parasitizing the host, but for many viruses in ants, this evidence is still lacking.
We are only beginning to understand the effects of viruses on ant behavior and physiology. Little is known of their pathogenicity because most infections remain chronic and asymptomatic. Indeed, the best studied viruses in terms of behavioral effects on ants are 3 viruses infecting fire ants: Solenopsis invicta virus 1, 2, and 3. The effect of these viruses span from slight fitness cost to colony mortality. Individual ants can be simultaneously infected with the 3 viruses at the time, or any combination. For other ant species we know little, but a recent study documented the clinical symptoms of acute bee paralysis virus at the individual and colony level of Lasius niger ants. Ants were fed bees that had been infected with this virus, and the acute bee paralysis virus was found to replicate within the ants. Forager ants presented impaired locomotion and were shaky with uncontrolled movements. The infected colonies were smaller due to having fewer emerging adults, and these acute bee paralysis virus are relevant for ant fitness because they may contribute to the weakening of colonies.
We suspect that the search for biocontrol agents will continue to provide a lot of information about viruses, their diversity, and their effects on ants over the next decade. Fire ants, Argentine ants, and other invasive ant species represent a major cost for both biodiversity and economies. These costs will effectively fund the search for future biocontrol agents. And viruses will remain a really central component in that search. But as Peter Flynn and Corey Moreau describe in their review of the paper, the most effective viral biocontrols will kill an ant colony quickly, making these the hardest viruses to find in nature, since the affected ant colony is often decimated before it can be sampled. We suspect that highly pathogenic viruses are likely to be hiding somewhere in the native range of both Argentine and red imported fire ants. Because these invasive ants are an international problem, and because highly pathogenic viruses and biocontrols are hard to find, we really promote the idea of a coordinated and multinational search for these biocontrol agents. If I was to dream the dream, the ideal would really be to have a facility somewhere in South America with hundreds of ant colonies that could be collected and cultured, monitored for mortality, and then sampled for pathogens. That facility could be funded by several countries around the world with samples made available to groups everywhere. That sort of coordinated approach could really advance the search for biocontrols. But also, our knowledge of the viruses in these ants would progress much faster than the piecemeal approach that we are currently taking.